Hello, I'm Sarah, and I'm excited to share with you alien algae. The secret scales you won't believe chlorodendrales are an order of green, flagellated, thecate, unicellular eukaryotes within the green algae class Chlorodendrophyceae. Prasinophyceae are defined by their cellular scales, which are composed of carbohydrates, and chlorodendrales are unique within this group due to these scales forming a fused thecal wall. Cells of chlorodendrales are completely covered in scales, which fuse around the cell body producing the theca, but remain individually separated on the flagella, of which there are typically 4 per cell. Species within chlorodendrales live in both marine and freshwater habitats, occupying both benthic and planktonic food webs. Additionally, they are photoautotrophs, meaning they produce their own food through the conversion of sunlight into chemical energy. Now, let's explore the fascinating world of where this unique algae thrives and how it interacts with its environment. Species within chlorodendrales are found in marine and freshwater ecosystems around the world, including locations such as the salt plains of Goa, India. Species occupy niches within planktonic and benthic food webs, in which all species are photoautotrophic and have an ecosystem and trophic role similar to land plants in terrestrial environments. Primary producers are consumed by primary consumers, such as zooplankton, invertebrate larvae, and heterotrophic protist species. Most species within this group are free-living. However, some species have become photosynthetic symbionts in animals. The animal species that can be in symbiosis with chlorodendrales is limited due to their need for sunlight for photosynthetic processes. Chlorodendrales populations in natural settings tend to experience extreme fluctuations in population numbers due to seasonal changes in abiotic conditions, such as ambient temperature, the amount of sunlight, and nutrient concentrations. This leads to what has been classified as blooms, which is the rapid increase in algae numbers during the spring and autumn months because of high light activity and the turnover of nutrients within the water column. It results from the high amount of sunlight and the mixing of the water layers, which resupplies the upper photosynthetic layer with nutrients, allowing for primary productivity to flourish. Now, let's delve into the fascinating details of cell morphology. Cell shape and size of chlorodendrale cells varies depending on the species. The cells range greatly in size from species to species, with an upper limit of 25 μm in length. Cells can be round, ovoid, elliptical, flattened, or compressed. There is great diversity in chlorodendrale cells. Now, let's explore the fascinating world of flagellar and cell body scales. Chlorodendrale scales are unique from other Prasinophyceae lineages because the scales fuse to form a theca, which acts as an exterior protective casing for the cell. All Prasinophyceae lineages produce these external scales within the Golgi apparatus and secrete the scales via the endomembrane system. Vesicles budding from the transgolgi face carry the scales to the cell surface, and upon fusion, release the scales to the external cell face. In chlorodendrales lineages, these scales fuse after secretion to produce the thecal wall, in which individual scales bind by cross-linking to one another. The flagellar scales and the cell body scales are structurally and functionally different, the main difference being the fusion of cell body scales. The scales also differ in size, shape, and macromolecular composition. The Golgi apparatus is only capable of producing one scale type at a time. Thus, species with multiple scale types must have different phases of thecal development separated by time and space. This also means that cell body and flagellar scale production occurs separately. Let's delve into the fascinating world of scales used for taxonomic purposes and species identification. The thecae of chlorodendrale species are often unique, and it is an important character for species identification and classification. Thecate features vary greatly from species to species, in which thecae can vary between 1-5 scale layers, in scale shape, in scale size, and in molecular composition/ultrastructure. These thecate features are genetically determined, and thus, they are a consistent and reliable characteristic that will not be influenced by environmental factors. Now, let's delve into the fascinating genera within chlorodendrales. There are 2 genera within the order Chlorodendrales, Tetrasomus and Scherphelia. Both species are green, photoautotrophs, flagellated, and thecate. The difference between the 2 genera is the presence or absence of pyrenoids. Tetrasomus contains pyrenoids, and Scherphelia does not. Flagella within the two genera are distinct in composition and morphology from species to species, which can be used to infer taxonomy and identification. Both genera produce three types of flagellar scales, in scale combination, and scale patterning is unique between species. Scales differ mainly in composition and ultrastructure, and these are the main features examined from flagellar scales when identifying species. The increase in knowledge about the morphological and ultrastructural features of flagellar scales, and the ability to identify species as a result, has been due to the development of advanced microscopy and staining techniques. Researchers have collected and analyzed molecular data from 13 prasinophyte taxa to better construct the phylogenetic relationship of early branching green algae. Phylogenetic analysis of the small subunit ribosomal RNA sequence was performed using distance, parsimony, and likelihood statistical tests. The analyses showed 4 independent prasinophyte groups, in which these lineages represented the earliest divergences among the chlorophyta. The most parsimonious tree created suggests that the Chlorodendrales lineage is a very early diverging group of core chlorophytes, of which there are 4 clades. However, the diverging order of this group remains unclear. Strange algae, with hidden scales, reveal a surprising secret.